We'll do as good as it was the second interview.
I hope I can match that, actually.
You have to.
Oh, thank you. I can feel the pressure, right?
No pressure. Well, thanks for joining from Basel. 1 hour commute is doable, no?
It's a long trip, yeah.
It's a long trip. So I will do the same. I'll do a quick intro about yourself. So you are general partner at Biomed Partners, so it's one of the most active VC or biotech VC in Switzerland. You're a very experienced drug developer. So you did, I don't know how many years at Roche?
22.
22, yeah. So, and you gained really a lot of experience. You told me on the phone you really pioneered some clinical proof of concept work. And then you were actually involved in the integration of Genentech. So we will basically talking about how to run properly a clinical trial, like evolution of translational medicine, and also what is it actually to transition from big Roche to now being on the other side of VC and helping startups run it. So didn't say anything wrong.
It's perfect so far.
And I saw actually, it's good, when I was searching for that image, I realized that actually Roche is building a second tower, which will be bigger than the first one. Should open soon, so yeah.
Yeah.
2 towers, yeah. Twin towers, I hope. Will not end the same way, but sorry, it was a bad joke.
Bad joke. It should be censored from the film. Right?
Censored from the camera. All right, so let's start with translational medicine. I mean, you told me you really pioneered like clinical proof of concepts at Roche. What do you really mean by that, and what do you mean by that?
Yeah, so thanks for that question. So basically, I think this concept started from my clinical fellowship, which I spent at the University of Leiden. And basically that was a concept where we were trying to very rapidly in early clinical trials to profile molecules not only for pharmacokinetics, which was those days the standard, but we were looking for early readout markers. So that is called in technical language PK/PD assessments. And I was those days, funny enough, although I'm Swiss, I was hired from a Dutch university to come to Roche. And they had generated those days a new department. You know, these big companies, they have a lot of changes all the time. So, but those days, the idea was to generate a department which was focused to conduct proof of concept studies. And I was hired into that department. And guess what? The second program I was working on was a molecule called bosentan, which ultimately became the lead product for Actelion. So those days, the researcher was a person called Jean-Paul Clausel, and so everybody knows him. It was a very intense period, right? And I was the young clinicians trying to do these first trials, and we could characterize the molecule and show some early efficacy signals. So that's the kind of work that we were doing those days.
But maybe can you add a bit of perspective to that? Like, why? I mean, was Roche really the first one to move in that space, or was
I wouldn't say that. So basically, maybe that's now with the perspective of 20 years industry experience, you see these waves coming, right? And so those days there was really a focus in early clinical readouts and all these new technologies. What was new those days, PK/PD modeling, all of that entered the space. We did what was called indication finding trials. The terminology proof of concept was introduced, and many groups did that, right? And so basically that was then further progressed with Roche's notion that you have to have better diagnosis, this concept of personalized medicine,
biomarkers,
which was then the next wave. And at some point, I became head of this department, which was in charge of clinical research and early development. And we run all these programs to characterize now not only the proof of concept, but to profile the patients. So to increase the likelihood of success by have better targeted therapies. So that's next to the wave. And if you see it from that perspective, probably what's now happening with the whole digital technologies, you get the next stage of better characterizing these assets early on with these new technologies.
Can you go a bit deeper in that? On how do you see digital playing a role in establishing this proof of concept?
I think the big change that you see compared to those days when we did the trial with, as I said, Bosenthal was my second project, and Roche felt there is no future, and the market is too small, and Jean-Paul was visionary enough to really build a whole company out of it, which tells you a lot about the dynamics between big pharma and biotech, but that's another question. So I think what you see now, it's really multidimensional, right? It's essentially, what you see now is that you really can get much better information on patient diagnosis. You can follow the outcome of therapies much better. There is real data coming in. So this is changing fundamentally the way we are going to go to do clinical trials. If you think about it, even this concept of phase 1, phase 2, phase 3 is something that was defined somewhere in the 60s or the 50s, right? Even that would need to be rethought, right?
What should it be now?
Huh?
What should it be now?
You see that regulators are moving in that direction, right? So you have conditional approvals for rare diseases where essentially you're going to go to provide outcome data only later on. I think it will be probably, if you spin time forwards, it will be a requirement that companies will have to provide a real data outcome from the market to reconfirm their label.
Yeah.
Right. So that will change the whole concept about what is confirmatory, right? Because so far we said we do a phase 3 trial, and then it has been shown the drug improves progression-free survival by 6.8 months, and that's it, and that's the label for the rest of your days, right? What will happen in the future is you will get real-life data back, and people will say, yeah, but under those circumstances it doesn't really work. You have to adjust it, that the whole focus is going to go to change from. But that's also a very important notion. These days, or in the old days, the pharma companies were in total control of the data sets, right? They could assess how good their drugs really were. Now in the new world, you will have real life data and constantly have feedback loops about what's really happening. And the ownership of those data is not only with the company anymore, it can be with insurances, it's with regulators, it's everywhere. Yeah. That will make things much more demanding for keeping drugs really in on the market.
That's good. Can you just place your mic a bit closer to your mouth? So on maybe can we switch a bit on translational science in general? I mean, we talked a bit about this, and like every country tries to foster more translation. I mean, I think here in Switzerland you're actually pretty good on that, but if you look at France, it's pretty much a disaster. They're really not good at this. But in Europe in general, we're not the best at translating science. So why do you see it? Why is it so challenging in your view, like to bring a molecule from the research to market? What's your take on that?
It's really interesting. I think even in Europe you have a lot of heterogeneity, which is probably cultural based, right? Yeah. I think fundamentally the difference with US is probably two things. It's the mindset of the academics, and probably also the availability of venture capital. I think the biggest challenge here, and I think it's probably in Europe, I still see Switzerland as one of the best places because we have really a lot of excellent universities, and the place is rich on innovation, which is actually coming out of these places, EPFL, ETH, University of Basel, and what have you. And there is a certain mindset in entrepreneurship in the academic space, and what is very special here in Switzerland is that you have access to experienced life science people from the industry. So that mix is making it easier here. But generally, if you go to other places, then you have, exaggerate, you have the professor who knows it all and owns the science and wants to continue his or her academic research in a biotech company, and that typically goes wrong. Yeah. And that limits it, right? I mean, if you just think if you go to Harvard or Stanford, then many of these professors, they at the same time have jobs in companies. So it's maybe also a cultural thing. It is in the US, it is seen as a prestigious thing if you are an academic and entrepreneur.
Yeah.
In many European countries, and partly even in Switzerland, it's kind of a contamination of your pure academic integrity that you start talking to business people.
Yeah.
And so it's all in this cultural space where you find the difference.
So for you, but Switzerland compared to other Europe, it's really like this link to industry is stronger than in other European countries.
I think that the Swiss are in Europe well off. I mean, other blades is Holland. I'm biased. I'm still teaching at the University of Leiden. So they are pretty entrepreneurial. Interestingly enough, the Danish. So what is also notable to your comparison with France, it's more the smaller countries who are more agile and entrepreneurial.
That's good. And what do you think about tech transfer offices? Isn't that the supposed to be the solution and creating the bridge and solving the whole problem?
That's really a mean question. No, I think that good tech transfer offices.
Who is in tech transfer? Tech transfer.
Good tech transfer offices are fundamental, right?
Yeah.
But I think they are a little bit. They are probably in a difficult situation, mediating between the intrinsic university administration and the need of entrepreneurial solutions and dealing with VCs and what have you. So I don't envy him for his job, but I mean, you see large differences. You see tech transfer offices who are really very good, smooth, cooperative, and others where you think actually they will never. I mean, their job is to block innovation. So you,
which ones?
No. When we have a beer, right?
Yeah,
that was really a mean question, actually.
Yeah, no, it's.
Can you ask some more nice things?
Being honest about tech. I mean, I think. At the end, they are very essential. I mean, the role they have is essential in bringing innovation to market.
But it comes back to your theme of the translation. I think that's really the fundamental. And what I'm fascinated is bringing research discovery into a clinical utility. I mean, scientifically, that's what always has fascinated me. But as a product, because essentially, and that's what we are still doing in biotech, because once you have shown it works clinically, the value is going up exponentially. And then from the whole business perspective, I mean, we were just doing this, our example that Stefan was mentioning, Alentis. It's a molecule which comes out directly of a research lab, and we are building a company. So this is
without
the whole, huh?
Without tech transfer office?
Oh, there are a lot of tech transfer offices in France, right? I think we're dealing with 2 or 3. Yeah. But so essentially you have 3 levels where this translation needs to happen for science, product development, and organizational, and that is a big challenge where which limits the use of this innovation in the different countries.
That's all right. And so on, maybe moving to clinical trial, you told me you're helping startups to design properly their clinical trials. What do you mean by that?
Sorry, to
design properly the clinical trials. What do you mean by designing properly a clinical trial?
it is, I think it's an art to design meaningful clinical trials. I mean, it's probably one of the privileges or benefits I had through my training, but also at the time at Roche, that we had really well-designed, challenged trials that were delivering the answers to the scientific hypothesis. Now, of course, one cannot compare this necessarily to every biotech company who is under much more financial constraint. But it's even more important for these companies to design their program in a way that you get answers, right? And unfortunately, you still see a lot of not well-designed trials.
Tell me more.
Yeah,
maybe for those who are running trials, like what's the biggest mistakes you see?
It is about the clarity of the hypothesis, right? It's about the realistic expectation of the endpoints. It is about the involvement of clinical people who understand these endpoints and the patients, and ultimately it is about the proper statistics behind, right? I mean, there are so many ways how you can introduce bias and invalidate your trials. And last but not least, very important is to understand your own drug, right? This PK, PD profiling. Many trials are run at the wrong dose. They are run at the wrong endpoint, insufficient powered. Not big enough. I mean, all the mistakes. I mean, you can join one of our courses at Leiden University, and I can tell you about it.
Tell us now. Give me some examples of really nice clinical trial failures in Europe.
No, I hate to talk about failures.
I mean, something that's public.
I mean, I give you an example which probably I'm still most proud of, where we turned the whole program around with a proper clinical trial. Yeah. There was a molecule which Roche licensed in. That was somewhere early 2000, end 90s. It was a BRAF kinase inhibitor. And basically, this molecule showed the first trial was just totally negative, right? Did not show anything. So without going into molecular pharmacology, but essentially, it was known that mutations of BRAF kinase are drivers of disease in melanoma, gastric cancer, and thyroid cancer. Right? So it was very disappointing that we didn't see anything. But when we transferred the program to Roche, I insisted that we transfer it, because basically the trial was not very good. Right? Because what they have done was they treated everybody with or without the mutation. And obviously, it's very difficult to expect an effect of a drug when people did not have a mutation, if you have a mutation inhibitor, right? So that was one problem. The other problem was, and basically they were probably put under too much pressure by their VCs or whatever, they had not optimized their formulation, so their formulation was very poor. So the drug was poorly absorbed, and the exposure that they had in that trial was impossible that you really could expect an effect. So maybe that's the poor story. I mean, this was a trial which from the very beginning was a total waste of money. We were treating the wrong patients with a formulation that never ever could be efficacious.
It was a VC-backed company.
Not all VCs are so brilliant. And okay, so then, what we did was obvious. We said, okay, now what we need is a diagnostic test to identify those people who have a mutation. And okay, that was Roche. We had to put the formulation department. They improved the whole thing, and we got to reasonable exposures. And then we ran the first trial, and it was most amazing, because you know how difficult it is to show efficacy in proof of concept drugs in oncology. But basically, the drug worked phenomenally. We had a progression-free survival of 9 months in the first trial. You could see the patient being cured because they had
So
melanoma, and they
from zero
disappeared.
results to 9 months.
So, I mean, this happens
With the same drug, basically.
once in a lifetime, right? But
Yeah.
essentially, you could argue that due to a poor trial design, under certain circumstances, that program would have been stalled, and it would never have been developed. And with a proper design, with the right questions, with the right profiling, with the right design, you can get an outcome.
Yeah,
As an example.
that's a good example. Maybe it brings a good transition to what you are now doing as a VC, and like obviously you're bringing experience in the startups you invest in, and you're helping them to design the clinical trial. Can you talk a bit more about that, on how do you actually help them? Why do you do it better than other VCs who are not that good, as you said?
No, there are many good VCs, basically. So Biomed, we have at the moment our third fund, so we are investing into the third fund. Why are we now particularly good? So I'm not sure we are better than others, but essentially, at least what I try to do is to find true innovation. I believe that ultimately only programs who are truly innovative have a high potential. Because if you see that the risk in R&D is high anyway, right? If you take something which looks more secure but is not so promising, even if it works, the upside is limited. Yeah. So you have to take more risk, go for the truly innovative thing, and then if that works, and sometimes it will not work, then actually it becomes truly exciting. So what we try to do is going for these truly exciting programs, and we have done some now in Switzerland, Amal Therapeutics in Geneva, was a very exciting technology. Tolremo here at the ETH, and just now Alentis. These are all very early programs with exciting science and exciting technology.
But can you help, formulate it otherwise? I think a lot of VCs don't have your background in really this 20 years of clinical research, and a lot of the VCs look more from a financial and like return point of view, and they have a knowledge about the industry, but they don't have really a knowledge on running clinical trials. So
maybe you can argue I'm not a typical VC, but basically I would argue that most VCs have very qualified people who can. The consortiums that we are putting together, the colleagues in other groups, they're typically very qualified people, right? But it's true. At the end, we should not be naive. A VC goal ultimately is to create returns, right? And the way we work in Biomed is that I'm probably more in finding innovation, designing the trials, helping people to set up R&D. And we have some very diligent experts who are then phenomenal in actually writing wonderful contracts that make everybody happy. Or almost everybody.
To talk with the founders. That's good. We'll open up for questions just after. And how did they manage without you?
I think they were reasonably successful. Yeah. Basically, otherwise the company would not exist. A VC who has no success is typically not long living. So Biomed 1, Biomed 2 were quite successful. And now we make Biomed 3 fund even more successful. That's the goal.
Never stop. That's good. One of questions. Same thing, don't be shy. Go for it.
Hi, I was very interested to hear your brief take on the computational side of things. I'm just thinking about all these. There's a rather heterogeneous group of companies that have become known as AI drug discovery companies. What's your take on them? What's their place in things?
Yeah, I mean, it comes back to the question about digital technology is changing the industry. I mean, it's obviously this happens also in pharma. I'm not necessarily a top expert on digital technologies, but I mean, it's obvious. I mean, in the chemistry space, I would say that by processing this information, you should be much more effective and efficient in finding hits, in predicting structure relationships and pharmacology. I would expect it makes finding of new molecules against new targets much easier and better, and probably more accessible also to smaller companies. Yeah.
So are you looking at to invest into AI?
I mean, fundamentally, I personally is my dogma in this. You should only invest into things which you understand. So if you want to invest into artificial intelligence, we would probably have to hire somebody who understands it. I don't understand it.
Makes sense.
Hope that answers your question.
There's a question? Yeah. You will be elected Mr. Questions.
With your experience in clinical development, how do you see the way forward to truly personalized cancer medicine, like personalized cancer vaccines? How would you then do kind of clinical trials in the conventional way, phase 1, 2, 3? If you have only n=1 as a medicine?
Yeah, I mean, this is a tricky question. So basically, as I said, I think some of the conventional paradigms will have to change because of all these new technologies. I mean, what you can do and has been done is that if you use certain algorithms to treat patients and compare that to another group that has been treated with conventional therapies, you can see differences. There are first trials who have been run like this, and I think that's also ethically possible. But personalized vaccines, it's a huge effort, right? Because you need to have the diagnosis, you have the algorithms, you have the CMC part. I think for your question, you probably would have to compare cohorts with and without this technology being applied.
Yeah, came in from. You had great questions, by the way. It's good.
With all these emerging technologies, do you expect that time to market for new drugs will become shorter in the future?
I mean, I was amazed to see about the FDA in recent years, right? As I said, now not to play the old man, but basically we had some quite tough discussions with the FDA when we went through some Roche programs. And I must see, at least what I have, maybe it's one of the differences. If you come as big pharma, they are tougher, and with biotech they are more open to discuss. I mean, we had a great experience with Polyfor, and Michael is here, and I was helping them as the CDO for a certain time period, and we run through FDA meetings, and I mean, I could call the FDA department head directly, and he gave me advice how to proceed. Right? I mean, imagine at Roche. I mean, first you had to call it in San Francisco somebody who is then actually approaching them indirectly. I mean, you were miles away from what's really happening. And so I must say, also for EMA, I mean, these agencies have today top people. They are educated. They are not the bureaucrats who are preventing innovation. They have very clear criteria. You can have an educated discussion with them. And FDA is seriously trying to make the access for new drugs easier. They have all their accelerated approval pathways. They are more than open if you go there for a pre-IND meeting. So they're on this avenue. So I think I think probably to your question, with the new technologies, they may be more open to get confirmatory data in a later step, because you can get those. In the old system, you got a license, and that was your license to market, and unless there was a serious safety issues, you could go on with it. So that's what I said initially. You have to expect a new world, where essentially there is a constant turnaround of validation of a drug's efficacy, and they have to do this just simply for the cost, right? Because people will not want to pay $100,000 for a cancer therapy that doesn't work.
Makes sense. Maybe to finish with one question, how was it for you personally, the transition to the VC side? Like, let's move.
To VC or out of Big Pharma? To VC.
I mean, to VC, yeah.
To VC, I must say it was not really planned. It was essentially, I mean, I think you need to keep an open mind about trying new things. I was approached whether I would be interested to join Biomed, and then I thought, okay, why not? I have never done VC. I don't really understand it. That's a good opportunity to learn it, right?
And so far, in 2 years?
I'm trying to learn, right?
That's a good conclusion. Great. Round of applause for Andreas, please. Thank you very much. Thank you.